Good morning. My project is protein function prediction using machine learning. And in the field of bioinformatics, understanding what protein do is a key step in knowing how living organisms function and in tackling new diseases at molecular level. But the traditional lab methods are of determining the protein functions are time consuming and they are expensive. So in this project, we aim to bridge that gap using computational approaches. We work with annotated biological data sets where the proteins have already known functions, and we apply supervised machine learning algorithms such as support vector machine and neural networking. First, in this project, we pre-process the protein sequence and extract meaningful sequences, things like amino acids length, and we use these features to train our model to classify proteins into their functional categories. And finally, we evaluate the processes by using standard metrics like accuracy, precision, recall, F1 score, etc. Coming to the content slide, we first begins with an introduction explaining the importance of protein function in understanding how the biological system works. And coming to the objective, our project objective is to pre-process protein sequence and from that extract meaningful features and apply machine learning techniques and evaluate their accuracy in predicting the functions correctly. And in the background of this project, we mainly focus on previous effort in the field of protein function prediction. And moving to the methods that we are used in this project, we use protein sequence data, extract amino acid frequencies, and train these models like random forest, XGBoost, and CNN to classify function based on their geotimes. And coming to the current status include the experimenting with multiple models where we achieve moderate accuracy, like exploring deep learning techniques to improve results. Proteins are the fundamental building blocks that carry out almost every biological functions, from catalyzing metabolic reactions and transmitting signals within the cell and defending against the pathogens, regulating gene expressions. Because of this, understanding what a protein does and their function will make us understand how each cell work and how diseases are generated. So in the traditional way, the scientists rely on the methods like gene knockouts, mutagenesis for determining the protein function. While these techniques are highly accurate, but these techniques usually slow or costly, and it is not feasible for the growing volume of the data we are having now. So the data sets like UniProt, CAFA3 are rapidly expanding with millions of protein sequences. To address these challenges, computational methods, especially this machine learning, have become important these days. By training models on known annotated protein sequences, we can make predictions about the functions of unknown proteins quickly at a large scale. And this helps the researchers to prioritize the proteins for further studies and to get progress in the fields like drug discovery, enzyme engineering, and etc. For the objective, we first began the data collection and then pre-processing. We gathered this protein data set from Kaggle, also from the UniProt, and then we clean the data by handling the missing values and preparing it for the model training by applying normalization, etc. Next, we focused on the feature engineering. It is one of the most crucial steps in machine learning. This involves extracting the useful features from the protein sequences and their annotations. Following that, we moved to the model development. Here, for now, we trained models like for random forest, XGBoost, etc. And in future, we are planning to do more uh, training data. And after training, we performed a model evaluation and optimization where we access the performance using common classification metrics such as accuracy, F1 score, recall, etc. This help us to understand how well the model perform, also how reliable this model can be for this project. The availability of large annotated data sets like UniPort and CFA3 machine learning approaches have rapidly evolved. Also, in initially simple model like KNN, K-nearest neighbor were common, but recent trend shows that a shift towards more powerful models like random forest and even deep learning models like CNN. A 2020 study using random forest on amino acid frequency features achieved about 30% of accuracy for predicting the geoterms. Also in the 2021, an SVM based model reported an F1 score around 40 to 50%, showing noticeable gains over these baseline models. Also, in most recently in 2022, a basic CNN model using one hot coded sequence, which means it's a way of representing the data like characters in a proteins or DNA sequence as numerical vectors that can be used by the machine learning or deep learning models, which reached an approximately around 45% of accuracy, demonstrating that the deep learning can effectively learn from the raw sequence data. The methods that are used in this project are: we begin by collecting the protein sequences in the FASTA format and their corresponding geoterm labels, which focused on molecular functions. After merging the data sets using their protein IDs, we filter the data to include only the top 10 most common geoterms to ensure the balanced data set for training. And then we moved to the feature extraction, where we calculated the relative frequencies of amino acids within each sequence and included the total sequence length as say additional features. These features were then normalized and the go terms are encoded into numerical labels and prepare them for the model training. Finally, we train the model using random forest classifier as well as XGBoost on this data using a 80 to 20 train split ratio, and the model performance was evaluated using accuracy, precision, recall and F1 score. And also to interpret this result, we also visualized some of the most important features influencing these predictions. Currently, we are working with the Uniport Swissport experimental subset as well as the CFA3, which include around 9,500 well annotated protein sequences. Each sequence is represented in a FASTA format, and annotation map each entry ID to their gene ontology terms, which is the geoterms, specifically focusing on molecular functions. Currently, we work two different models. That the first one is the random forest classifier with 30 trees and a depth of 10, which achieved an accuracy of approximately 24%, and an XGBoost classifier which improved the parameters that slightly increased the accuracy to 25%. We can see that the XGBoost model shows better F1 score for certain geotypes, as we can see from this slide. Also, this evaluation was done using standard matrices like a precision, recall, and F1 score, which gave us insight into where the further improvements are needed. This is the code review for the random forest, and these are the libraries that we are using in here. This pandas is used for handling the tabular data. This bio.sqio is used for parsing the FASTA files, and this sklearn is a tool that used to for model training and the evaluation. And this matplotlib is used for plotting feature importance. And this snippet of code the shows that we are loading the FASTA file. This is the FASTA file that we are using in here, which is included in the CFA3 dataset. This record.id.split extract the uniplot entry ID from this FASTA header. And coming to the labels, it reads a text files containing mapping of protein ID to their GOTEMs. This is the text file that we are using here. and this filters the geoterms to keep only the molecular function categories, and they finally merges this protein sequence and their GOTEMs using the entry ID. And now each protein has its sequence and its geofunctional annotation. And coming to the feature extraction code, uh, this the given string contains 20 standard amino acid, each represented by a single code, and this function, extract feature function, takes a protein sequence as an input and returns a dictionary of features extracted from this sequence. And this uses a dictionary comprehension to calculate the normalized frequency of each amino acids in the sequence. By doing so, the result is 20 numerical features are normalized between the values of 0 and 1. And here we add sequence length as an additional feature. This gives the model some idea about the protein size, which may be functionally relevant for this project. And we apply the functions here. We are applying the functions to all the sequences. Then coming to the another code of snippet, we elect okay here we elect the top 10 most frequent geoterms for simplicity and balance in this case, and this use label encoder function to convert the geoterms to their numerical labels. And in this code we are training the random model classifier using the arguments n estimator and maximum depth. This n estimator is equal to 30, which means we are creating 30 decision tree. Here higher n estimator means increasing the accuracy, but while doing so we also increase the training time. And the max depth, which we are set to 10, which means that tree depth is limited to 10, which basically means that the number of splits a decision tree can have, which helps prevent overfitting and reducing training time. And here we are using the evaluation for this project using the classification report, which generates a classification report which shows precision, recall, and F1 score for each the geoterms. And this code basically plot the confusion matrix heat map for the result. This is a confusion matrix heat map. This confusion matrix basically is a table used to describe the performance of this classification model by comparing the true label with its corresponding model's actually predicted label. Here the labels we are using is the actual geoterms. And for example, as we can see in the geoterm label here, the 70 value, it has a 70 value, which means that 70 proteins were truly labeled as this geoterms, but they were incorrectly predicted as geo 003700. If our model have a higher accuracy, this will the all the darker cells will be aligned in diagonally, which means that the model works perfectly. But in this case there is little bit of misclassification, as we can see that the confusion matrix is somewhat scattered here, because of the we only got the accuracy of 24%. And similarly for the code for XGBoost, we only change the XG classifier function. We use the n estimator and max depth and evaluate the model, which gives us an accuracy of 25%. In summary, we developed a sequence-based learning framework to predict protein functions using features derived directly from the primary structure, specifically the amino acid frequencies. Also, by applying these supervised models like Random Forest and XGBoost, we achieved encouraging results that demonstrated and the potential of these models in identifying these molecular functions. Also, this approach not only improves efficiency but also improves the scalability, making it very usable for larger bioinformatics field. Also, the broad impact of this work spans to multiple areas like drug discovery, enzyme engineering, functional genomes, where rapid and reliable functional annotation of protein is increasingly critical. This slide presents the key features that supported our project. First, we used data from the CAFA3 datasets, which is a well-known benchmark for protein functional prediction. It provided a valuable collection of annotated proteins sequences that help train and evaluate our models. And secondly, we refers to the research involving neural network and random forest models in protein function prediction. And these studies give us the information into different machine learning techniques, their architecture, and their previous works. Finally, we used a UniProt database which served as a foundational source for sequence and annotation data.